Hello, class. So, today we are going to continue chapter number 10, that is Biotechnology and its application. In today's class, we are going to cover one session and one subsession of that session of NCERT, that is session 10.2, and its subsession 10.2.1. So, basically, in today's class, we are going to study about how biotechnology has done a lot of revolution in the field of medicine. So, let's go ahead and we will see today one of the most important example of like how biotechnology has improved the medicine field. One of its example is insulin. So today we are going to see the this technology, how this technology has done a major changes in the healthcare. Disclaimer. So what are going to be today's learning objectives? So what we are going to learn from today's class? First, to explain the significant impact of recombinant DNA technology on healthcare. Today we will see how this recombinant DNA technology has played a major role in healthcare, and how it's introduction of this recombinant DNA into some gene has improved our aspect of medical science, and we are using it today as medicines. Second, we will see to explain how genetically engineered insulin solved the historical problem of insulin sourcing for diabetes. We know that some diabetes patient has to take insulin, okay, to maintain its sugar level in their body. But but what used to happen before, how do we get insulin? So we used to get insulin from the slaughter pancreas of slaughter pig, and but there were so many side effects of that. We will talk about that, what was the side effect and all. So how this genetically engineered insulin came into picture and changed the whole dynamic of this, we will see today in today's class. Then we will understand the production of recombinant human insulin. Then we will see how this recombinant human insulin has been produced. So let's go ahead. This is going to be index of our today's class. First is we will cover biotechnological application in medicine, which is 10.2 session of NCERT. In this, we will cover one of its subsession, that is genetically engineered insulin, which is session 10.2.1. Then we are going to summarize all the points. Then there will be some NCERT based question which we are gonna see or solve. Then there will be some keynotes for you guys. And lastly, there will be some interesting questions that you guys gonna solve yourself. So first of all, what is recombinant therapeutics? As the name suggests, recombinant therapeutics, it says the therapeutics in which we have used recombinant DNA technology. We know what is recombinant DNA technology. We have studied it into deep detail in previous chapter. So what is a recombinant DNA technology? So it's a technology in which we can get our desired gene. We can get a desired trait in host or whatever you say in that. So what is recombinant therapeutics? So therapeutics, the therapeutic field in which we use the recombinant DNA technology. And in this we can produce a lot of therapeutic drugs too. They do not induce unwanted immunological responses. So, what is the benefit of using this recombinant therapeutics? So, one of the major importance of this using this recombinant therapeutic is that first of all we can get our desired property, whatever we want, whatever desired gene we want. There should not be any undesired gene or anything. And the main importance is that they do not induce unwanted immunological response. So for example, as I was saying that initially what how to patient with diabetes used to get their insulin, so they can extract their insulin from slaughter pigs or cattle. But what can happen, this insulin which we get from some other organism act as a foreign part can act as a foreign particles in a body. And what happens when a foreign particle invades your body? There will be a immunological response. So he- so in this recombinant therapeutic, what happens, there won't be any unwanted immunological response. They are more safe and effective as compared to others. 30 recombinant therapeutics have been approved for human use the world over. So in the world, there has been 30 recombinant therapeutics that has been used, that has been being approved for human. In India, 12 of these are presently being marketed. So among the 30 recombinant therapeutics which we have all over the world, 12 are 12 of them are present in India and has been marketed. For example, so this recombinant human insulin. So this recombinant human insulin is used to treat diabetes and is marketed in India too. Second is recombinant factor 7. So this recombinant factor 7 this used to treat hemophilia. So now what is now from now onwards we will study what is genetically engineered insulin. So before that we should know what are the properties of insulin. Insulin is a proteinaceous hormone, the hormone which is made up of protein. It is synthesized by pancreas. So insulin is been produced by and synthesized by pancreas. It helps in regulation of blood sugar level. We know that insulin has been used to regulate the blood sugar level. So this is a picture which represent that how insulin and what is insulin, and how this pancreas release insulin. So this is an pancreas, and this pancreas is responsible for releasing this insulin hormone. So what is like need of this insulin? Adult onset diabetes could be managed by taking insulin. So what is this adult onset diabetes could like? So there are two diabetes. One is insulin resistance, and one in which there are it's not been like insulin resistant, but in that there is like insulin can't be produced. So for this adult onset diabetes, what happened, we can manage this diabetes by taking insulin, because in this we have not only not been resistant to insulin, but our body has stopped producing insulin. So we will take insulin from outside, so it can manage our body glucose level. In the absence of human insulin, it would be delivered externally, as I said. This led to extraction of insulin from other animals. So if a person is not able to produce insulin in his body, so how we can take it externally, we can take it from outside, but we have to extract it from other animals. What are those animals? So usually, l- previously it used to be pigs mostly. So what are the problem which is associated with conventional insulin? Earlier, insulin was extracted from the pancreas of slaughtered cattle and pigs. Earlier, what used to happen? This insulin was extracted, was taken from pancreas of slaughtered cattle and pig. Slaughter cattle and pig means cattle and pigs who has been killed to get this insulin from their pancreas. It caused some patient to develop allergy just because this insulin is not from, like, it's not its own human's own insulin. So it can, what can it lead to? It can lead to, and it can develop some allergy. As I said, in some patient what can happen? So that patient body will treat this insulin as some foreign particle, some foreign protein. So what happen if the body recognize some foreign protein? It will develop a immuno it will trigger the immune system, and there will be some immune reactions, which is not good. Now talking about human insulin, what is the structure of the human insulin? So the human insulin, a mature human insulin, is consist of two short polypeptide chains. So there are two polypeptide chains of insulin. One is called chain A and other is called chain B. It is synthesized as proinsulin. So what is proinsulin? So usually what happens, a mature insulin is consist of chain A and chain B, but in human it is like synthesized at proinsulin as in, along with chain A and chain B, it has chain C also. And to make it a mature insulin, we have to remove that chain C. So this is a human insulin. It look like so proinsulin and mature human insulin. So how it is, like, this is chain A and this is chain B. This chain A is consist of 21 amino acid and this chain B is consisting of 30 amino acids. And this chain A and B has been joined together by disulfide bonds. This is the mature human insulin which can be utilized by the body. So if this is proinsulin, so insulin synthesized as prohormone, as I said. Proinsulin contains, if that was mature insulin in which there was chain A and chain B, so what is actually prohormone? Proinsulin. So this insulin usually was synthesized as prohormone. Initially it produced as prohormone, and this proinsulin contains what? An extra C peptide along with chain A and B. So this proinsulin need to be processed to get mature and functional hormone. So now what we have to do to make it, as we said that a mature insulin has two chain A and chain B. A consists of 21 amino acid, B consists of 30 amino acid. Both are joined together by disulfide bond. But this proinsulin, which is actually the body synthesizes insulin as prohormone, this proinsulin contains an extra C peptide. And to make this proinsulin into mature insulin, we need to first process it. So how we will process it? By removing this C peptide. So during the maturation, what happens? The C peptide is removed, as I said. The mature functional insulin consists only of chain A and B. These two chains are linked together by disulfide bridges. So this is the NCERT figure in which maturation of this proinsulin into insulin has been shown. So this proinsulin, like, this is peptide A, blue and green is the peptide B, and in middle this in middle this gray part of is the C peptide. So to make this, and these are these disulfide bridges. So to make this pro-insulin into insulin, they did, they removed this C peptide. This C peptide became now free, and this A and B peptide has been linked together by disulfide bridges, and this is how now mature insulin looks like. So now we know what is the normal structure of insulin. Now we know how this first insulin is synthesized as proinsulin. Now then we have to remove the C peptide to make it as mature insulin. And we also know that initially what used to happen, we have to take insulin from slaughter pig and cattles. But after this, all this is something that we till now know. Now we will talk about this recombinant insulin. Now we will talk about how this recombinant insulin has been made, and how has been it changed the picture of this insulin, use of this insulin in medicine. So first of all, what was the recombinant DNA challenge? The main challenge was getting chain A and chain B assembled into a mature functional insulin, because we know that this insulin has been first synthesized as proinsulin. So the main challenge was how we will get that chain A and chain B assembled into a mature this insulin, functional insulin. So what happened, there was a breakthrough which happened, which this Eli Lilly, this is an American company, they did it in 1983. Now what they did, because they know that if we will take gene from mm, if we have to synthesize this whole proinsulin, then it will become very difficult to do that pro processing step where we have to remove this C peptide and all. So what this company did, this is an American company, what they did in 1983, this company developed a method to produce human insulin commercially. And what they did actually, so before that, you should know that the recombinant functional human insulin, they market it as Humulin. They given the name Humulin, and there is a picture showing that how it looks like. So this is the figure how it looks like. It came in. So what they did actually, so for we will study each and every step of that. What was the process of production of recombinant insulin? So what was the process of production of recombinant insulin? What this company Eli Lilly did? So first what they did, so they did this DNA sequence preparation. They take this two separate DNA sequence were prepared. So this was their strategy. They won't synthesize this C peptide. They will take the DNA sequence for synthesizing this A peptide and B peptide, and later join it by disulfide bonds. So then there will be no role of C peptide. So then there will be at last a mature hormone. So what they did, they took that two separate DNA sequences were prepared. One corresponds to A chain and the other is for the B chain of human insulin. This point should be noted because from this slide only you guys can answer NCERT exercise 5. Question number 5. I mean to say that. So those DNA sequences were prepared from the human genome. So these sequences were prepared from the human genome only, that can code for A chain and B chain. So this is how it looks like: DNA sequence preparation for recombinant insulin and transformation of E. coli. So this protein has been expressed by E. coli. We have used E. coli as the host. So what we did a take a vector. In that vector there is a promoter, there is a B galactosidase. This is a selective marker which we can use if you guys remember. Then there will be DNA sequence for insulin A chain. There is also part of this ampicillin resistance gene which is used in selective marker thing. And for other thing, DNA also, what they did, same thing, promoter, this selective marker, and insulin B chain DNA sequences. Now what they did, they insert it into a host plant, which here is E. coli, and after that we can get our desired protein. So now step 2, first they have, as I said, first they have prepared this DNA sequences, then they did this vector insertion. Two sequences were introduced into two plasmid. The two sequences, one for the chain A and other for the chain B, were introduced in two plasmids. Those plasmid were then put into E. coli to produce insulin chain. As I have told already, like how this, they take this two sequences, insert it into the vector, and then express, take E. coli as host and expressed, and then produce the insulin chain. This will produce insulin A, chain A of insulin, and this will produce chain B of insulin. And from here, this you guys can answer the NCERT exercise question number 9. Now, what are, like how we produce it? Inside E. coli chains, A and B were synthesized separately. Okay, so you guys can see. So as you guys can see, this is a vector with this DNA sequence for chain A. This is another vector with DNA chain B sequences. We insert it into our host E. coli. Then we will see that which E. coli has been transferred or not, transformed and not, that we can see because we have this selective markers, beta galactosidase gene and even ampicillin resistance gene. Then what we will do, we will culture this E. coli which has been transformed, means which are been able to produce this chain of insulin. After this culturing, we will purify this beta galactosidase insulin fusion proteins, and then we will take chain and A and B, and then there will be some step for purification of this chain A and B. And then what we will do, by using this, not using this, like by then they are being joined by using disulfide bonds, and then there will be a active insulin which can be used in healthcare. Okay, now which can be used for the treatment purpose and all this thing. Now this insulin is ready to inject into the patient who needs the insulin. Extraction and assembly. The two chains were extracted first from the bacteria, as I said, then they were combined by creating disulfide bonds to form human insulin. As I said, key now, by this culturing of the cell and then purification of this fusion proteins, and then we will purify this chain A and chain B, and then we can create disulfide bond to form a human insulin, and this human insulin is now ready to be taken by patients. So what is the summary of today's class? So we have first seen that how this recombinant DNA technology can revolutionize healthcare by enabling mass production of safer therapeutic drugs. In today's class, we have specifically seen that how initially how we have to slaughter pigs and cattle, and then we extract their insulin, and that can cause some side effect. First it can bring some allergy to the patient, or then some patient can treat it as foreign materials, and if the patient will treat it as foreign material, then the immune system going to activate. Once immune system going to activate, then it is going to kill its that insulin, that foreign cells, which is now not safe for the patient. So then what happened, then this recombinant DNA technology came in. Eli Lilly company, what they did, this is an American company, and that company did what, they used their brain, because we know that Insulin first synthesized as proinsulin. Okay, and this proinsulin have chain A, B, and C, but we didn't want the C peptide. And we can't and what is this challenge? We can't even synthesize this whole protein ki, okay, we will synthesize this insulin, proinsulin, because this E. coli don't even have the machinery to post process this insulin. So they don't have this machinery to remove this C peptide and all those things. So this was a challenge. So how this company Eli Lilly has overcome this challenge? What they did, they have created this. They have created this. They didn't create it, means they have extracted this chain A and chain B individually and then created the disulfide bond and then make it as a mature, what do you say, mature insulin. And how they did it? By using recombinant technology, recombinant DNA technology, using vector. They have, in first vector there was DNA sequence for chain A only, then in the second vector there was this DNA sequence for only chain B. Then all those, when they, that vector get injected into the host, now that host has produced this are the chain A and chain B respectively. Then we can see like how they are, which of that E. coli has been transformed or not. Then using cell culture and those downstreaming process and all those things, we can extract chain N and chain B, which by creating, then by creating a disulfide bond in between them, we can make a mature insulin which can be used. So, genetically engineered human insulin solve the problem of limited and immunogenic animal sourced insulin for diabetes. Now, this genetically engineered human insulin, what it did, this they solve this problem because we can initially we has to have limited insulin, and even it was like immunogenic animal sourced insulin, because as I told, we can even have this immune, can trigger this immune response because of these things. Now human insulin is synthesized as prohormone, requiring removal of a peptide for maturation, that we know. Now this human insulin synthesized as prohormone, in which there is chain A, chain B, and a chain C peptide, chain C also, and we need to remove this chain C for the maturation of insulin. Eli Lilly pioneered this recombinant human insulin production by doing what? By separately synthesizing this chain and A and B, and this combining chain A and B. And how they synthesize from? So they synthesize using this recombinant technology, and this E. coli usually express this proteins. So this is the summary. Let's go ahead. So now what does question 1 says? So question 1 says, before the advent of, question 1 says, before the advent of genetically engineered insulin, what was the primary source of, insulin for diabetic patient, and what were its drawback? So the question is asking us, before we have this discovery of genetically engineered insulin happened, what was the primary source from which people, diabetic patient, used to take their insulin, and what was its side effects and all? So let's see the answer. So the solution says the primary source was pancreas of slaughtered cattle and pigs. Okay, what was its drawbacks? That some patients develop allergies and other type of immune reactions to the foreign animal protein occurred. Supply was not always sufficient. So as I said, as I said, that what was the drawbacks when we used to, when a patient used to take insulin from the slaughtered cattle and pigs. First, some patient can have the tendency to develop allergies. Second, some patient can treat this animal protein as foreign material, and this foreign material going to trigger the immune response. And even the supply was not always sufficient. So, second question, what was the main challenge Eli Lilly faced when producing insulin using recombinant DNA techniques? And how did they overcome it in 1983? So, the question asks, what are the challenges, what are the problems which this American company Eli Lilly faced when they were producing insulin, and how then they recover, how did this overcome this problem, and in 1983 they have come up with this recombinant insulin technology. So let's see. So the main challenge was getting the insulin assembled into a mature form, that is functional form. How they overcome it? What they did, they prepared two separate DNA sequences corresponding to the chain A and chain B of human insulin. Introduce these sequences into plasmid of E. coli to produce the chain separately. So what they did? So as we know that what they are facing, that how they will assemble this insulin and into a functional form. So what they come up with the idea. What was that idea? That they will prepare two separate DNA sequences. One DNA sequences that is going to produce chain A of human insulin, and the other which is going to produce chain B of human insulin. Now, so for that, what they did, they introduced these sequences into plasmid of E. coli to produce the chain separately. This is the NCERT figure only, which is showing that how this blue part is A peptide, green is B peptide, and this gray is C peptide, and how they have to get rid of this C peptide to get a mature fully functional insulin. Then they extracted from the E. coli, they have extracted the separately produced A and chain B. Then they combine them in vitro by creating disulfide bonds to form mature human insulin. So as we know that what they did, now they have extracted this chain A and B separately. Then in vitro means in laboratory, in outside the living organism, what they did, they have created this disulfide bonds between this A and chain A peptide and B peptide and make it as a fully mature functional human insulin. So what are the keynotes of today's class? So the keynotes of today's class was that the first commercially available recombinant human insulin, that is Humulin, was approved by the FDA in 1982. That in 1982, FDA is the Food and Drug Administration. This is the organization who give approval to all the drugs and food and all those things that can be used by people and that can be marketed. So, the first commercially available recombinant human insulin, which this Eli Lilly company has produced, which is called Humulin, was approved by FDA, means the Food and Drug Administration, in 1982. The discovery of insulin in 1921 and 22 by Banting and Best and colleagues was a major medical breakthrough, saving millions of lives before this recombinant DNA technology. So before this recombinant DNA technology has been discovered, has been, you know, in 1982, before that in 1921 or 22, these two people, Banting and Best and their colleagues, were the major, they brought a major medical breakthrough, and you know they have this did the discovery of this insulin and all those things before this DNA recombinant DNA technology. E. coli is a preferred host for recombinant protein production due to its rapid growth, ease of multiplication, and cost effectiveness. Why we use this E. coli as a host? Why? To produce this recombinant protein. Why? Because of its rapid growth, ease of multiplication. We know that every 20 minutes E. coli can divide and multiply. So ease of multiplication, and it is cost effective. Now there are some interesting questions that you guys gonna solve yourself. So first question says, what is the main benefit of recombinant therapeutic drugs over those from non-human source? Means this question is asking that advantages of recombinant therapeutic drugs over what? Over this non-human sources, like from animals and all those things, plants. Now question 2 asks, what is the C peptide and what happens to it during insulin maturation? So we have studied a lot of time in this class, like what happened to C peptide. It got removed, but you guys have to answer it. Question 3 asks us, why can't insulin typically be taken orally? So why we have to administer insulin into body like intravenously? Why can't we take insulin as a oral, like why can't we take this as a normal medicine? Why can't we take it as orally? You guys have to answer that. Question 4 says, what specific microorganism did Eli Lilly use to produce insulin chain? This is also we covered in today's class, that which microorganism the Eli Lilly company has used to produce this insulin chain, to produce this chain separately, and then you know that we create this disulfide bond. Thank you. So here are some daily practice question from this biotechnological application in medicine. Let's solve this. So what the question number first says, which of the following is a reason why insulin isolated from pigs or cattle may cause allergic reaction in some patients? So this question says that what could be some reason, that what could be the reason by because of which this insulin that we have isolated from pigs or cattle, slaughter pigs or cattle have caused allergic reaction to some patients. So let's see the options. So options are, first, the insulin is not properly processed into mature form. Okay, let's see the next question. The insulin contains the C peptide. Third, it is a foreign protein. And fourth, it cannot form disulfide bonds. So the main reason why this body can, body can react and cause and have this allergic reactions from this non-human source insulin. So that one of the reason could be what? It is a foreign protein, and because the body will treat it as a foreign protein, it is going to develop some immunological reactions or response against it, and this can cause some allergic reaction to the patient. So the answer is C, it is a foreign protein. Second question asks, in the production of recombinant insulin using E. coli, why were chain A and B produced separately? So they are asking that in this recombinant technology where we have produced this insulin, why we have produced chain A and B separately? Why can't we produce it together? So let's see the options. What are the options? The first option says E. coli cannot process proinsulin into insulin. Okay, let's see the next option. The C peptide interferes with insulin function. Okay. The third option says the disulfide bonds form spontaneously when chains are combined. And the fourth option says E. coli lacks the machinery to combine the chains. So, what could be the answer of this question? So the first option will be the answer. Why? Because this says that E. coli cannot process proinsulin into insulin. As I told that this E. coli don't have any machinery or something to do this post processing steps, so it cannot process proinsulin, the insulin which have peptide A, B and C, into insulin which only have peptide A and B. So this is the answer. Why the other options can't be answer? The C peptide interferes with insulin function. It's not about insulin function. Because of this C peptide, it will be a pro-insulin only. It won't become an insulin. So the third option says the disulfide bond forms spontaneously when chains are combined. No, it is not like that. Third, that last options in this, which is E. coli lacks the machinery to combine the chain. E. coli lacks the machinery to remove that chain B, that chain C. So the answer will be option A. That E. coli cannot process proinsulin into insulin. So next question says, why is gene therapy for ADA deficiency involving lymphocytes not a permanent cure? So this question is not from, like, you know from today's class, but still we will answer this question. So this says that, why is gene therapy, we know what is gene therapy. Gene therapy is a technique in which you can, by using this recombinant technology and all those things, we can, you know alter the gene of the patient, and you altering that gene can you improve the health or some of that patient, if that gene is responsible for that disease. So there is a gene therapy for ADA deficiency involving lymphocytes, not a permanent cure. So this is ADA, this is an enzyme which is responsible for this immune system to work properly. So if deficiency of that, what the deficiency of that cause disease or cause a disease, and because of this deficiency we can use gene therapy as the therapy. But even using this gene therapy, it was not act as a permanent cure. So why it not act a permanent cure? So basically what we do, we take the lymphocyte and we just did the gene therapy with this lymphocyte. But the reason is that this lymphocyte is not immortal, means that lymphocyte also die. So once this lymphocyte who has this genetically altered gene, in which this ADA gene is present, it will also die with some time. So that's why we can't cure it permanently. So I understand that you guys not gonna understand all this concept in today's class, but we will answer this question, and in this next class I think we will study this. So the options are, lymphocytes are immortal cells. No, as I said, it is not a immortal cells. Then lymphocytes have a short lifespan and required repeated infusions. As I said that this therapy has been done, this gene insertion and all those has been done to lymphocytes, and lymphocytes have a short lifespan and they required this repeated infusion. That's why it cannot be permanently cured. It has some other, if we will do this therapy to the bone marrow, then it will be can be permanently cured. That things we will discuss in next class. Third options, the ADA gene is not correctly inserted into lymphocyte. No, this is not the reason. Even inserted normally function, even inserted properly, we know that lymphocytes don't have that large lifespan, and after some time that lymphocytes also gonna die. Bone marrow transplantation is more effective that it is, but it is the reason why this gene therapy for ADA deficiency involve lymphocytes, not a permanent cure, is the answer is this. Save and next. How does PCR aid in early diagnosis of infectious disease? We have studied PCR in the last chapter. This is what is PCR. PCR is a polymerase chain reaction. So, how this polymerase chain reaction can be used to diagnose an infectious disease? So, what actually a PCR do? So, this PCR has this potential to amplify the gene, or a particular gene of that, or DNA fragment of that organism or something. So if we have a infectious, so how it can help in diagnosis of infectious disease? First, it increase the concentration of antibody. No, it has nothing to do with antibodies. Second, it amplifies the pathogen's nucleic acid for detection. Yes, it can do that, because we know what is the actual role of PCR. It amplifies, it multiplies this, you know this multiplies this DNA, and so it can, what it can do, it can amplify, it can multiply the pathogen's nucleic acid for detection. Third option, that it uh, identifies mutation to autoradiography, no. It measures antigen antibody interaction, no. What it does, it amplifies this pathogen DNA or RNA, which is nucleic acid, for detection. And even if we don't have symptoms of that disease, because even if we have smaller amount of this nucleic acid of that pathogen is present inside our body, we can detect it through PCR. Last question, what last question asks? Last question says, in ELISA, detection of a pathogen is based on. Now, what is ELISA? ELISA is enzyme linked immunosorbent assay. So, what this enzyme linked immunoassay can, how it can detect pathogen? We know that it actually works on this thing of uh, antigen and antibody interactions. So the antigen of the pathogen and the antibody that has been created, that has been produced by. Okay, I okay, wait. Okay, so time was up for this because I was explaining it to you, but it's okay. We will see the answer of last question also. We can see that this all four options are correct. Let's see five options. So we were telling about this enzyme linked immunosorbent assay. So what it does, that it can detect, it is based on this antigen antibody interaction. And what this antigen antibody interaction will do, because we know that the pathogen has this antigen, and because of that antigen, our body has the ability to produce the antibody against them. So this binding of in this interaction of antigen and antibody can be used to detect pathogen, and this is something based on ELISA. So this is the last question. Okay, so yeah, thank you.